special supervisor t-shirt.
From Microbe TV, this is Q&A with A&V. Today is December 21st, 2022. I'm Vincent Racaniello, and joining me tonight from Chevy Chase, Maryland, Amy Rosenfeld. Hello, Amy.
Hi. Hello, Vincent. How are you today?
I'm very well. It's a little bit warmer today. It's still cold, but what is it today in New York? It was, let's see. Let's take a look. -1. A little warmer than yesterday. How about down there in Chevy Chase?
So that's about 30, right?
Yeah.
Was that the high today, at the height of the day, or is it -1 right now?
No, it's -1 right now. And actually, It's in Scotch tape. I don't know about Manhattan. How about you?
It's 0.
Cool.
And guess what today is? So today is big day. It's the solstice day, right?
Yeah, it's
shortest day of the year.
Yep, big day.
And it's 4th night of Hanukkah.
Happy Hanukkah.
Yeah, more donuts. I don't like donuts.
Let's welcome our moderators. I saw Steph. Steph, almost forgot. How could you forget? I'm so hurt down to the core of my being. Actually, just kidding. Just want to get a reaction. Steph is here.
Don't be guilty. What are you? Don't give her guilt. We're not doing stereotypes.
Les is here. Vanity is here. And Tom is here. Thank you all for joining us to moderate. And welcome, everyone, to your Wednesday night, midweek Virology corner of the internet. And we're happy that you're here tonight. And we're going to try and get through many of your questions. But before we do that, I have a surprise photo. I didn't even tell Amy this. It may not show up properly. Well, let's do this. Are you ready? Okay. Can you see that, Amy?
I see. It looks like little Vincent and his mom.
So, in fact, yes, this is me down here. We'll put the thumb in the mouth. I don't know.
Well, of course.
Maybe a few months old. I don't know. You got to like the hat, right? And then above me is a picture of my mother when she was very young. It was an adorable dress, right? Cute mom. Kind of face looking like mine, right?
Yeah, except your eyes must be your dad's.
Why do you say that?
Because the eye doesn't look anything like your mom's. So by definition, it has to be the other parent.
Yeah, I suppose. Anyway, I just wanted to show folks that. So that's that. You see the frame is quite old also.
Both the frames are quite old, but they're super cute. I had a frilly dress like that. Does she have ruffled underpants on underneath with white shoes and ruffled socks?
But look at the shoes with the laces all laced up. That's pretty cool.
Yeah, right? Does she have ruffled socks and white tights?
It looks like she has tights, but I don't know. Anyway, that's just a little thing there. You can see I'm taking a picture with my Twiv phone. What I really wanted to talk about today are these two, one article. Let's make sure that we can all see this. Okay, so this is the New York Times. Amy sent this to me a few days ago. New COVID booster shots cut risk of hospitalization by half, CDC reports. And as soon as I saw this, what did I say, Amy? I don't believe it, because they just want to push the booster. So let's go over it. Reducing the risk of hospitalization by roughly 50% compared with certain groups inoculated with the original vaccines. Okay, there are a number of problems with this. I want to go through with you. All right, so here is the original paper. Morbidity and Mortality Weekly Report. Early estimates of bivalent mRNA vaccine effectiveness in preventing COVID-19 associated hospitalization among immunocompetent adults aged 65 or older. This is a New York State network, right? The A&V network, I believe, is mainly New York State. Yes, multi-state. Oh, no, it's multi-state. Sorry. Okay, 73% additional protection compared with past monovalent mRNA vaccination. And you should already look at that and say, wait a minute, we're comparing a current regimen to a past regimen? That can't be right. And it's not. They didn't compare it neck and neck with a ancestral booster. Bless you. Bless you, Dr. Rosenfeld.
Thank you.
Okay. So they say here, everybody over 65 should receive a bivalent booster, and you've heard Paul Offit talk about it. And so here, this is from September to November. Adults over 65 admitted for COVID-19-like illness, and it's not clear if for or with, and that's one of the things that Paul Offit always talks about. And then they said, what kind of vaccine did they get, and what's the story? So a couple of things that are very important here. The median interval between receipt of the booster dose and illness onset was 29 days. Now, Dr. Rosenfeld, what's happening at 29 days after a booster?
High antibody titer.
Very high antibody titer. And so when compared with unvaccinated patient, vaccine efficacy defined as hospitalization of a bivalent booster and prevented hospitalization was 84%. Okay, that's with unvaccinated people. When compared with patients who had received 2 or more monovalent-only mRNA doses 2 months or more before illness onset, the relative vaccine efficacy of a bivalent booster was 73%. When compared with patients whose last dose was 6 to 11 months and over 12 months before illness onset, that's the monovalent dose, relative vaccine efficacy 78 and 83%.
So let me get this straight. We're saying that if you go and I look at you 29 days after you get a booster, 84% of the people are protected.
That's right.
From going. But if I then compare you to people who haven't had any vaccination for 12 months or more, then you're still higher.
It's 84%, yeah. So it's a booster, right?
No,
But they're comparing them. They're comparing.
But what you're comparing doesn't make any sense.
Well, of course. And the problem is this is comparing the present time with people who were vaccinated before. You can't compare the two. So they note all the limitations.
Not at different time lengths. You have to ask what happened after that first boost 29 days later.
The correct experiment
would have done to compare the first boost 29 days later compared to
and at the same time
the booster at 29 days later.
Wait
a whole year. You can't
not do an observational trial, but rather have two groups. You get bivalent booster versus a regular booster, because I would argue, first of all, this is so close to the booster. Yeah, you're going to get antibodies protecting you. In 6 months, it's going to be a different story. And secondly, I would argue that the ancestral booster would do exactly the same thing.
Well, that's what I'm saying.
Exactly right. There's zero evidence that this effect has anything to do with the bivalent booster, which they're pushing, and they're going to say, we need a new booster every 6 months or whatever. And that's just wrong. That's just wrong to conclude from these data. I hope folks get this, right? They do point out the limitations. Because use of the monovalent vaccine as a booster is no longer authorized, this analysis could not compare the effectiveness of a bivalent with a monovalent. Right. So you could just use the original booster if you want to boost people. Anyway, so that's that. I asked Paul Offit what he thought. I'm sorry. I didn't show you that quote here. I want to bring that back up. Here's the limitations. Because the use of the monovalent as booster is not authorized, they couldn't compare them. So I would argue that at best, this shows you that any booster would work. And within 29 days after getting the vaccine, it'll reduce hospitalization if you believe the hospitalization data. So I asked Paul Offit what he thought. Paul is always nice. Here's what his reply was. Of interest, our hospital tests every child who is admitted for COVID. Currently, there has been an increase in the percentage of children who test positive, consistent with an increase in cases right now in the U.S., but only one child in our hospital now is actually being treated for COVID. The rest were admitted with COVID, not for it. Nonetheless, all are reported as COVID hospitalizations. So many problems with comparing bivalent boosting now with historical monovalent boosting, different times in the pandemic. Often those who chose monovalent boosting earlier than those who waited for the bivalent were older or had comorbidities, and it's not surprising that boosters boost antibodies for a limited period of time. Hard to watch the way this is played out. Okay. I thought you would all be interested in that. All right, let's get rid of that. So I'm not a big fan of this headline saying, it's 50% better. You're 50% protected because maybe any booster would, or maybe this number's not even right because of the way they're defining it by hospitalization. Okay, Amy, is that good?
Yeah.
All right, let's see here. Well, let's get to our questions. Happy solstice, everyone, from Billy Bob. There you go. Oh, so Amy, we did a doggy podcast with Nels.
Yeah, the great, the German Shepherd and everything. Yeah,
it's very good. It's really cool. I really liked it very much, and some people here were there.
Eventually, you'll get to the bananas.
Two old school friends are still sending me articles on anti-everything, feeling really bad for Fauci. I don't know if he feels badly. I think he understands what's going on, right?
I think he's done.
Apparently, it's getting very cold throughout the U.S. A lot of people are talking about that. So let's get past that. Okay. Anyway, welcome everyone, all 245 of you. And let me ask you at this moment already, please hit the like button. Should have on Tweevo, Amy, the other night, we had 80 people and 78 likes.
What two forgot to hit the like button?
Amy doesn't say, good job. She said, who forgot to hit the like button? So let's go, folks. Hit that like button. It helps people find us. Here we have
Wasn't a negative response.
No, I'm just joking. Hi, A&V. My relative is 7 days testing positive. His partner just tested positive. What are the chances of him being reinfected before Christmas gathering or passing it on, even if
Zero.
He tests negative?
Zero
reinfected, but Christmas is only a few days away, so that person could be shedding. I don't know what the vaccination status is.
I doubt he's shedding. If he's been testing positive for 10 days, I doubt he's shedding.
Well, the 7-day one, yeah, but the other one just tested positive. So,
but Christmas is still 5 days away. I doubt he's shedding.
Yeah. Anyway, if you're worried, wear a mask. Correct?
Sure, absolutely. Always err on the side of caution.
So we have Happy Hanukkah and Merry Christmas and Happy New Year coming up, right?
And Kwanzaa, right? Isn't
Kwanzaa.
that the day after Christmas or
anything
something?
Else?
I don't know.
So here's another one from Greenfields. Can flu vaccination lessen shedding of influenza, I presume, if infected? I believe it does blunt the shedding. It doesn't eliminate it. But
it just reduces it.
Yes, here's a good sentiment. Thank you, mods, for helping make the best night of the week possible. That's very funny. The dogs. Here we go. Last week, A&V highlighted the RSV FC paper in Amy Paper's Washington University study. FC antibody function required for vaccine protection in COVID. Amy is simply amazing. Uncanny. Amy, you're uncanny.
That's what I heard.
Do you got any canned things to open up?
Like what kind of canned things? I don't eat canned vegetables. I don't eat canned fruit.
You don't eat, there's no cans of anything in your house?
There's a can of hazelnut paste.
There you go. How about evaporated milk? I bet you have a can of evaporated milk.
What would I do with evaporated milk?
It's used for cooking, baking. No, and if in a pinch,
it could be. It's just not something I normally have in my house. Like condensed milk is super sweet, and I don't usually have it in my house.
But the
I don't even have a can of tuna fish because that is kind of yucky.
If you run out of half and half, you could use the evaporated milk.
No, if I run out in the half and half, I get in the vehicle in my pajamas and I go to Target to get my half and half because I'm so pissed off.
Is that the closest place for you to get half and half?
Well, there is a Whole Foods around the corner, but sometimes they don't have half and half. They're not reliable.
All right. DGH writes, A&V, NIH funding mRNA vax to deliver a primer hemagglutinin, readying the body's B cells to react quickly if faced with flu. How could this work against all strains? Amy, how could it work against all strains? You put all the hemagglutinins in, right?
You put in all the hemagglutinins, or you target a conserved region.
Yeah, I mean, the one that Scott Hensley is doing is...
Yeah, but there are others. Yeah, but there are others.
Well. Yeah, so Scott Hensley said he's going to Europe for the holiday. He'll be back next year to do TWIV.
Well, you only got one more TWIV of this year, and it's Friday.
Yeah, so next year is in... Well, no, the following week is the 30th, right? That's another TWIV.
Yeah. Yeah.
Okay, Kang. Hello, Kang. Does Shingrix protect against chickenpox? Well, it's the glycoprotein of the virus, so it should, wouldn't it? I mean, it hasn't been tested to protect against chickenpox. It's been tested to protect against shingles. And he says, if not, does the virus change while sequestered in nerves? No, as far as we know, it does not change. So I would assume that if you got it and you'd never had chickenpox, it would prevent chickenpox. But I don't know because it's not been tested for that. Okay. Turbo JV says, nice dog. Should we be worried about all infections in China producing a new variant? No. I don't think there are that many. Are there, Amy? How many are there in China?
I don't know, but it's drama and a half there.
What's the source of the
both
drama?
The Wall Street Journal and the Times are reporting disasters, like, that they had this lockdown, people protested, now they're trying to release the lockdown, blah, blah, blah.
Look,
it's a disastrous policy.
I think it's been worse than any other place that's been hard hit. We don't know where the variants come from, frankly. And,
did see the paper from the South Africans. They had to retract it.
I saw that, yes. And Christian Anderson said, I could have looked at this and told you it was wrong before they even submitted it. And
so did the RES. So did Ari in England say that too.
It's too bad because it was Felix Drexler who was on TWIV. They should have put it up as a preprint. They should have put it up as a preprint.
Yeah.
And you know what's weird? I'm sure the referees said this, and the bloody science editors who, make the decisions, they decided not to pay any attention to it. Why? Why did they do that? Is there something wrong with science, Amy?
Can I plead the fifth?
Arama? Is it an S-arama?
It's beyond a shitarama.
Somebody
I don't know what's beyond a shitarama, but it's like the black hole of shitaramas.
Someone said, Greenfield says, Amy has such a great smile. Can you smile for us? Did that hurt?
No.
Anyway, I wouldn't worry, particularly about China, because there are a lot of cases in many other places as well.
For sure.
Oh, look at this. Times Art Studios. The Green Times Art Studios. That's a good name. My Virology Fix. You bet. You bet it's your Virology Fix. Yeah. Yep. All right. We're looking at the pictures. Amy, behave. What did you do that was not behaving, Amy?
I don't know, but I really don't like comments like that. It's not necessary to write.
Sean is a good guy. Sean's a good guy.
No, he's not. I'm not telling him to behave. I think it's rude.
I'm sorry.
And I don't think you should read these comments. You don't.
Sorry.
And the thing is none of these comments are ever directed towards anybody else.
Yeah, they can be directed at me.
No, but they're not.
I'll find
None
one.
of these comments are ever directed at you.
My apologies. I'm sorry. Again, I made a mistake.
It's not you to apologize.
No,
I
picked it. I thought it was funny that he said he's being behaved in a funny way, but I can understand that you would take it wrong.
There's no wrong about it. There's no right or wrong about it.
Dr. Graham Lloyd Jones says the COVID virus can reach the lungs through bleeding gums directly through the bloodstream. Hence the important. There's no evidence for this at all, is there, Amy?
Not that I'm aware of, but oral health is very important just in general for heart health.
Yeah. There's zero
It's
evidence
just
that
in
the virus
general.
gets anywhere through the bloodstream. Zero. Vanity says, I have the same frame. My mom found it in an antique store. Huh. Yeah, well, in my frame, that's my mom, of course. Any comment on Dr. Sette's new paper indicating multiple vaccinations infections is of no real benefit with T cells? Well,
T cell epitopes are conserved.
yeah, there's no need. We're going to have Daniela Weisskopf on TWIV in January.
Cool.
And she
Cool
is a
beans.
collaborator of Sette, so we'll talk about it for sure. You should listen. Yeah, by half compared to what? That's the thing. Old information. That's the problem. How can we know that a particular vaccinated patient would have a worse experience getting COVID if they weren't vaxxed? We could presume that, but it would be impossible to know, right?
Yes. It's impossible to know unless it's impossible to know. And even if they had COVID before they got vaccinated, you really can't make an extrapolation.
Well, I would say that if you look at the effect of vaccination on severe disease, and it has an impact, if it's 80% effective against severe disease, then you can say
You can say on
overall,
average.
yes, on average, right. You can't
But
say
you
for any
can't
particular
pinpoint
person.
for an individual.
Right, but for
It's
this
just an
person
average.
who I think is trying to argue that we shouldn't vaccinate, it's no, on average, you're going to get less severe disease with vaccination. There's no question about it. Amy and Vincent, Victorian study revealed molnupiravir and Paxlovid need to be given within 0 to 3 days of illness. Earlier, the better. Are rapid antigen tests up to the diagnosis? Well, it depends on how that study was done, right? Was it done using PCR or antigen tests? The question is, Amy, compared to the time you're positive with PCR, how much later are you positive with an antigen test?
A couple of days.
So that could impact the efficacy of molnupiravir and Paxlovid, couldn't it?
Yes.
That's a good point.
But
But
the thing is it does any antiviral, whether or not you give the flu antiviral.
Yeah, but I think the studies should be done using the antigen tests, the less sensitive ones, to see if you can still treat early enough to make an impact, don't you think, instead of PCR?
Well, obviously you didn't have a PCR test. You just had an antigen test, and you took Paxlovid, and you said it made an impact. While you are one person and it's anecdotal, I'm assuming that the majority actually fall in that category nowadays.
That's a good point. So on Friday I started sniffling and throaty stuffy, right?
Throaty stuffy? Yeah, sure. Throaty
symptoms
stuffy.
on Friday, and they got worse on Saturday. And then I started Paxlovid on Saturday, and that was fine because Sunday most of the symptoms are gone. So, yes, it's an N of 1, but
Dixon was also the same, so it's an N of 2.
Yeah. Hans says, last TWIV was excellent. They could predict with amazing accuracy when and where yellow fever cases would appear in Brazil. Is there a reason why this can't be done for Zika dengue? Well, they were doing it based on the monkeys getting yellow fever, right? They were watching the virus transmit through monkeys, and so you can't do it for Zika. Zika, as he said, there's not really a known non-human primate reservoir for Zika. But there is for dengue, right, Amy?
Yes.
There is a non-human primate reservoir, but I guess in theory it could, but there are plenty of human cases all the time, so I'm not sure you need to do that.
Well, the reservoir also has to develop symptoms, right?
Yes. Well, you just go test them randomly. Just take blood or whatever. Sue says, I'm with you on the hospitalization stats, apples and oranges, and I can attest minimal COVID admissions here in Canada. Okay, I have to take your word for it because I can't attest to it, right?
Right.
Here's Greenfields. Are we any closer to discovering longevity of T B cell defense with COVID. March 22 was my 4th vaccine, no history of infection. Wondering if by year 1 I'll need a top-up.
Is this like when you put gas in your car?
Yeah. Yeah, but gas in my car. Did I tell you how much it costs to fill my tank?
Yes,
It's
I do not
ridiculous.
want to discuss it again.
Oh,
I had to scrape you off the ceiling.
So how long are the key is how long are memory cells going to last? The memory T cells and the memory B cells, how long are they going to last? And, you can predict that it's going to be 5 to 10 years. For SARS-1, it was 17 years T cell memory, right?
Yeah.
So you just have to keep looking at the same cohort of patients every year and see how long it lasts. Otherwise, I'm guessing it will be at least 5 to 10 years, right? Oh, by the way, I meant to show these. So here's our microbe TV stickers, right? Look at, I got a big stack of them. I got 2 stacks. Oops.
Well, now you're black.
Yeah, I hit the wrong button. I got 2 stacks, and actually I have 3. So if anybody wants these, I filled out the rest of the envelopes last night. Send me an email, Vincent@microbe.tv. If you haven't already, if you already have, don't send me another one because I'll just send you more, which is not bad, but.
Well, maybe they have more than one computer. Or maybe they have more than one iPod.
Yeah, I automatically.
iPad or whatever. Maybe they need more. Maybe they need them for in the office door or the office window. There's lots of places that they could put them.
I still have trouble stepping on your speaking, Amy. I don't know when you're done. Remember Margaret Thatcher? They always used to interrupt her in Parliament because they thought she was done speaking. Anyway.
Fine, not Margaret Thatcher.
I send out 2 to everyone, but if you want more, Vincent@microbe.tv. Here, I even have a little thingy here so you can see.
Wait, where's your Spike shirt? You didn't say anything about your Spike t-shirt campaign. You got to say your Spike t-shirt campaign. It's getting close to when everybody should be buying them, putting them in their stockings.
Oh, I should have.
For the next person.
I should have worn
we need their Spike t-shirt campaign.
Long sleeve.
Where's
Spike
the button?
T-shirt. Oh, Maron, I'm going to get it. Just chill, man. No, that's the Amy papers. Well, you should look at the Amy papers.
No, we need the Spike t-shirt button.
All right, I'm looking for it here. Where is the Spike T? Vaccinated.us. There we go. It's kind of small.
Well, make it bigger.
So, all right, Vincent@microbe.tv for stickers. The Amy papers I post all the papers she sends me and Daniel every couple of days, every day basically. Look at Amy's drinking a Cosmo.
No, I'm not. I'm just drinking cranberry juice.
And vaccinated.us for Spike t-shirts. So till the end of the year, if you buy a shirt from vaccinated.us, use the promo code MicrobeTV. They'll give some of the profits to Microbe TV, and we can use that for our science education. This is tremendous, folks. So if you haven't bought one, and the cool thing is you can send a gift card, so there's no time for shipping, but you could still buy someone a shirt on a gift card and send it to them by email, and they get it. And I've done that to a bunch of people. So I'll leave that up there for a few minutes. Okay, back to the questions. Sally says, so basically they didn't control for time pass booster. No, they did. They just compared it to an ancestral booster, and you can't. You have to have 2 groups, one with a bivalent and one with an ancestral to see if they're different. I mean, what if the ancestral booster gives you the same results? You have to bother making a bivalent booster, right? And you don't have to change every year either, because I think that's the key, that they're going to think from these bogus data that we have to change every year, but not. All right. The bivalent booster is EUA. This provides great immunity from liability. Well, I don't think there are any issues with that. It just doesn't do much compared to the ancestral. All right, on multiple TWIVs, Rich mentions a lot of people illustrate poxvirus structure incorrectly. Do you know which model is most correct? I will tell you. He has a website. What is it called? Poxvirus structure, Florida, I think. Let's see. Poxviruses. All right, I don't have it here. I'm going to have to ask. He has a site where he's got the correct structure. So the sticky, yellow sticky. Put down that I have to find out the right poxvirus structure, please. Hayden says, thoughts about the suggestion that increased SARS-CoV-2 prevalence in China could give rise to more dangerous variants, which could be a problem
We
for
just did that question.
Yeah, we just did that. No, I mean, they're no more than any place else, okay? There's plenty of circulation everywhere. Yes, for bivalent or monovalent for those at risk 4 to 6 months after the low. Yeah, if you want to. So, in my opinion, to pull off, it said this: if you're 75 and older, you could get a booster, and it's the only thing you can get in the U.S. is the bivalent. So, get it. But he also thinks the right thing to do is have your 3 shots or 4, and then be ready with a plan like Paxlovid. Don't take a boost every 6 months. That's not a good public health plan. Ariel says, how long would it take to develop a new COVID vaccine, mRNA vaccine, if a variant that escapes immunity is found? I think it would take, how long, Amy? 6 months or less?
Sure. Yeah, sure.
Because all you need to do is make the mRNA, which they have a scale up for, encapsulate it, and then do a little bit of safety testing. Maybe it'd take 3 months. Not a long time, less than a year. Is the key to fighting viruses T-cell activity? Well, it depends what you mean by fighting, right? If you mean
sometimes
preventing serious disease, then often it is, yeah. Right, Amy?
Sometimes. Yeah. Doesn't seem to have a role against preventing serious disease for enteros.
So for enteros, it's antibodies, right?
Yeah. Neutralizing antibodies are the best biomarker for protection against the development of severe disease.
Not FC antibody portion? No, it's neutralizing antibodies.
Well, we've never looked, but traditionally it's thought that it's neutralizing antibodies. But that may change as my lab expands and does some research.
This is Amy's alley, right up Amy's alley, right?
Yeah, this is my baby. This is my important virus. I work on important viruses, you know, Vincent.
Mm-hmm. Okay.
I like to tell myself I work on important viruses. How's that?
Okay, here's one I cannot say anything about. Dr. Lustig says the countries that did the best during the pandemic were those who eat whole foods, not processed foods.
Yeah. I don't know what a whole food is, and I don't know. Like they went to the Whole Foods market?
No, just not processed. So you get broccoli and you cook it yourself, as opposed to buying a tray with cooked broccoli in it, right?
Sure, fine.
However, this kind of an analysis is fraught with confounding errors. There are other things that could be having the effect. So I don't think you can conclude that without. If you're just going to do observational studies, you can't conclude that. You'd have to do some experiments.
Hey, I could do observational studies that said everybody who didn't eat fish on Friday did better.
Yeah.
Or anybody who just ate chocolate and not potato chips did better. Or eat potato chips and you'll do better. Who knows?
Tess wants to know if poliovirus can affect cotton rat brain cells.
Define a cotton rat brain cell. What is a brain cell?
Any cotton rat brain cell that you could find in a cotton rat skull.
Well, it infects the motor neurons and the motor cortex at the back of the brain.
Yeah, cotton rats were used to passage the Lansing strain initially. Did you know that, Dr. Rosenfeld?
Yes, Charles Armstrong, 1928, 99 passages. Although he could have just waited for us to poop it out, and then he could have just taken some of our poop and put it into the brain and found it much easier. But yes, but it doesn't infect like the sensory neurons in the cortex or the hippocampus or the pituitary gland, right? It affects the motor neurons in the motor cortex at the back of the brain and the medulla. So you have to define what brain cell you want to talk about.
Okay, but at least there are some brain cells that can be infected in a cotton rat brain.
Yes, I just want to say that there are some,
All right, I
but there
just
are
don't
also
want you to
some
pick.
that do not.
Just
So
so you
you
know,
got to be specific.
no, Tess is not a scientist. She just wants to know. You got to be forgiving,
Yeah.
right? Ian, thank you for your contribution. Last week you mentioned pembrolizumab. It's a monoclonal. I know a patient who took it. It was phenomenal. The melanoma shrank significantly. Is it based on virology principles? No, it's just an antibody to a tumor marker, right, Amy?
I believe so.
Pembrolizumab. Let's look it up. Pembrolizumab. Keytruda. Yeah, it's the checkpoint inhibitor.
Yeah, it's the PD-1.
Yeah, it's a directed against PDL1, and this allows the T cells to be awakened when they go into exhaustion, right?
Yeah.
Yeah, it's. I don't think viruses had anything to do with that. Do you know, Amy? I don't think so.
No, they didn't.
I'm gonna highlight some happy comments now that don't have questions. I so happy to see you two. Your podcast is my favorite. Merry Christmas. Thank you, Janie, very much. Really
Very
appreciate
nice.
it. And
How
the
about the
green
holiday season?
the Green Times Art Studios is in Taos, New Mexico. Okay, Green Times Art. I used to go to Taos a lot for science meetings. And what's that restaurant in town? The Taos Inn, right? I love that place. Very expensive, but it's good. Ryan tested positive 3 weeks ago. He was negative 7 days later. My partner is now testing positive. Should I be okay, or could I catch COVID again so soon?
I'll be fine.
No, you won't catch it again. Nope. You don't have any canned beans, Amy?
No, I don't like beans.
Amy, you sound much better this week.
Yeah, I feel better.
Is your tonsillitis gone?
I still have a little cough from like the lung infection that resulted. It'll be fine.
Here you go. Amy has such a great smile. So sometimes I pick
That
the
is
right
true.
comments, okay, Amy?
I see.
Sorry about that.
It's okay.
Rob, thank you for your contribution. Happy festive fungus. Happy festive fungus to you as well.
I think that that's a play from the Seinfeld, where the guy had made up his own holiday, like Festivus or something or other. Somebody will tell you about it. George's dad made up his own holiday, Ben Stiller. I think it was Ben Stiller. Maybe it wasn't.
Festivus is a holiday celebrated on December 23rd as an alternative to Christmas.
Yeah, but it was famous from the. It's famous from the Seinfeld episode.
Oh, it is? Okay. I didn't know that. Anyway, thank you, Rob, for your contribution.
I know because of Andrew. Yes.
Vaxman says, if you got a bacterial infection and died, would you be disappointed? I don't think you can be, right?
How if I'm dead? How do I know?
No, I would be disappointed to die, right? Especially before my time. What is your time, Amy? How old should you get?
I don't know. When I decide that it's time, I'll let you know.
I'd like to live to my 90s. That would be good.
Well, that would be good.
Which would be
That would be fun.
Between 20 and 30 years more.
Okay, that would be fun.
That
would be fun.
And I would like to be healthy. So on Twin, this week in neuroscience the other day, we were discussing we actually reading emails, and one was about Alzheimer's. And they said there are 3 ways to really hedge your bets against Alzheimer's, Amy.
Okay, now have shitty genetics.
No. 3 ways. First, exercise. Second, get good sleep. And third, use your brain. Think all the time. So I got 2 out of 3 covered. I got the exercise, I got the thinking, but I don't have the sleep covered. So
well, you'll get better at sleeping.
Oh, you're so confident.
What makes you think I'm not confident and supportive?
I'm going to get better at sleeping. Am I going to figure something out? Anyway.
Yeah, you'll be less stressed. You'll figure something out. You'll go to sleep earlier or later. You'll rearrange how you sleep. Maybe yeah.
I think
You're not an insomniac, a natural insomniac like me, so I don't think I need to worry.
You're a natural insomniac?
Apparently so.
Who
I've been
told you
in
that?
my mom, I checked it out. She
Uh,
said, "You
Mother's
sucked at sleeping when you were a kid, Amy.
you sucked at sleeping. That's great.
Not as bad as your sister, but you were not a good sleeper." My youngest
Uh,
sister used to like to watch before until she was 3. She used to like to watch The Love Boat at 2 a.m. That's when they showed reruns.
Love Boat. Okay.
Well, what can I say?
All
She
right,
likes.
Amy. Sorry, I interrupted you. Go ahead.
That's
Finish.
fine. Doesn't
Amy, you're
matter.
right. A can of condensed milk is only good for taking space in the cupboard. I have it, and when I run out of half and half, I use it. Okay.
It's too sweet.
Evaporated milk, I mean, not condensed.
Well, why would you ever run out of half and half?
Well, like I did this morning. I didn't go and get
Really? You
any.
ran out of half and half so you didn't have coffee?
Well, I had coffee, but I put heavy cream and a mixture of heavy cream and milk into it, right?
Well, that's basically half and half.
Yeah, it's half. I guess I don't know. Anyway, 350 people are here, and that's probably good because it's holiday week. But 211 likes. So hit the like button, please. We would like to.
What were the other 140 likes?
Hey, listen. I tell you what. If you don't want to hit the like, give us some money. Hit the contribute button. It's a good
Hey.
trade off for the Christmas week. Give us some money for our education efforts. At Like Greenfields, thank you for your contribution. €5. That's the euro
That's
sign,
very
right?
lovely.
That's cool.
Yeah, it's very
Amy,
lovely.
do you have any canned tomato paste? Yeah, there you go. You must have canned tomato paste and canned tomatoes. That's how you make tomato sauce.
That's maybe how you make tomato sauce. It's not how I make tomato sauce.
Polio Pete. Hello, Polio Pete.
Yeah, where are my cookies, Polio Pete? Where are my Pignolia cookies? I don't see no cookies.
Polio Pete will bring them another time. Okay?
Where are my cookies?
Hey, Amy, take it easy on Polio Pete.
You don't understand. If you play with my cookies, you're not playing nicely. Cookies are very important. They're like a food group.
Did you hear my picks on last TWIV?
I
Dr.
did. Your
Rosenfeld?
pfeffernusse things and the Pignoli cookies.
Well, let's pronounce them correctly. Pignoli
Pignoli
and pfeffernusse.
and pfeffernusse.
Yeah.
Yeah, I heard.
Really good.
Because Grandma used to make them. Although I'm not really clear how the German got into the Italian family, but I heard it all.
It's a holiday cookie. And
Yeah,
Amy
and
has
then
made those for me in the past.
I have.
And they're really good.
I have.
Anyway, I love your handle there. It's great. I think you told us the story, but I don't remember. If you want, you can repeat. Anyway, any association between viruses and gluten intolerance, celiac disease? I'm not aware of any. Are you, Amy?
It's being investigated.
So it's more on the order of microbiome that does stuff, right?
No, it was a pushed forward idea by Kate from Harvard that we talked to. She did IBD and she found echovirus RNA in the gut.
Yeah.
But she could never find any additional virus.
Okay.
And we told her not to publish it, but she published it in Science Translational Medicine, and then she moved from Harvard to Moderna.
So enterovirus infections are associated with the development of celiac disease. That's the study probably you're talking about. Yeah.
Yeah. It's kind of like piss poor.
That's Amy's assessment of the work. It's pee poor, right?
Yeah, pretty much.
Reverend Sasquatch is here. He's off this Wednesday and catching it
That's
live.
lovely.
Thank you. Love Dr. Wilson on Twiv. He was good. Really good, yeah.
Debunk the funk.
I'm working on getting on
Malcolm
Gladwell's pod. I'm not working on it. His agent actually called me and said, would you be interested in being on it? And I said,
Gotta
sure.
be on Malcolm.
She said, what would you talk about? I said, well, isn't that your job? I said, but I could talk about a lot of things. So I pitched her a bunch of potential topics. So that would be fun.
You could talk about technology and science. You could talk about viruses. You could talk about biology, neurology. Gosh, what a disaster.
Charlotte
What can
says
you talk
China
about?
still has the least amount of COVID deaths in the world. Yes. Well, they did an amazing job early on, right? With the
Sure, we
lockdowns.
can go. We can go with that.
Very severe lockdowns. And speaking of China, how is the supply of antivirals in China?
I don't know.
I don't know. I don't hear of. I mean, the molnupiravir and remdesivir are U.S. products, and they early on decided they didn't want our stuff. Right? They made their own vaccines. Now they're making their own mRNA vaccines. So I suspect they're making their own antivirals, and I haven't heard of any being made. But let's look it up. China, COVID, antiviral. That's the best way to do this.
Google me, man.
China approves its first homegrown COVID antiviral. I remember this. This was from this summer. It is a repurposed HIV drug. So it's the first oral antiviral in China.
Okay,
And
sounds good.
It's a polymerase inhibitor, Amy. So there's at least one EN.
Okay, sounds good.
Nava's question is, if the FC is important, can commercial monoclonals still be effective if the tests indicate that they're not? If the neutralization tests indicate that they're not? That's a very important word to have in there. Okay, here's a little anecdote. One of the monoclonal cocktails used to treat Ebola virus disease is a mixture of 2 neutralizing and 1 non-neutralizing antibody. And they found that the non-neutralizing antibody was important for the efficacy, the clinical efficacy. So that tells me, yes, non-neutralizing functions are important. So even if you have a non-neutralizing antibody, it could still be important. To do that, you need to have an animal model to test it in, right?
Sounds right.
You good with that, Amy?
I'm good with it.
This is a good question, Hat. Could the cumulative effect of yearly boosters enhance the risk and or severity of myocarditis? So as you know, the highest risk is in young men.
Young men. Yeah.
So yes, absolutely. Each immunization has a risk of myocarditis associated with it. And so that's a good reason not for all age groups, right? But it's a good reason not to just boost indiscriminately, don't you think, Amy?
I do.
Polio Pete is on the roll tonight. He's got another one coming up, too.
Wow.
He's just trying to make up for not showing up. Well, he had a good excuse, but...
Well, I no see no cookies. He could have sent the cookies without him. I'd be fine with that.
Them. He's got to deliver them in person. Come on. It's okay.
I would be fine with 2 deliveries of cookies.
You want me to send you some cookies?
No.
Here's another question from Polio Pete. What type of evidence would the scientific community need to prove a vaccine injury? Well, we do have one vaccine that causes injury, and we know it. It's established at a rate of 1 in 1.5 million doses. Right, Amy?
Yes.
The oral polio vaccine. And the...
Well, we also had the RSV vaccine that caused severe disease.
Yeah, RSV disease vaccine, they gave it to kids in a trial in the 60s, and then when they went out into the world and got infected, they got worse disease than the control group. So that's a very clear association. So when you have clear associations between the vaccine and some effect, paralysis, more severe respiratory disease, and the numbers are good, and statistics support it, and you have control and vaccine groups, that's the kind of evidence that you need. And so
not the made up kind.
Yeah, not the made up kind. And we are very aware of that. So the Vaccine Adverse Events Reporting System, VAERS, collects everything that people throw at it. Oh, I got a vaccine, and the next day I threw up. Right? People can put.
Yeah, I got a vaccine, and the next day I had a car crash.
And so they go through all of those and they decide which ones are valid or not. Most of them get thrown away because they're not valid. One incident is not going to establish any association. Hello, Laura and Kip.
Yeah.
Thank you for your contribution. Merry Christmas, Happy Hanukkah, Happy New Year from the two PharmDs and their daughter, who happens
Laura, Kip, and Emma.
All right, so come by sometime and visit. All of you can come visit the incubator. You just have to let me know when you'd like to come, because most days I'm there and I'm potting away or editing, and it's a lot of fun. I can give you a cool tour, and I can tell you stories. Right, Amy?
Yeah.
So come. Charlotte says, is there any evidence that the whole food diet will benefit? No, there's no evidence. To get evidence, you have to do a clinical trial. You need to do a randomized controlled clinical trial, which means you have two groups. One gets the treatment, one doesn't. And most of the time, people don't do that. So that's why if you do an observational study, we'll say, oh, in this town they eat this, and in the other town they eat this. And here in the first town where they eat it, they have a difference in COVID. That's no good. It's an observational study, which has a lot of other things could affect the outcome. We call those confounding factors. So not a good way to prove causation. Hello from Brazil. Hello, Raphael. I just mailed your stickers today because I recognize your name. I'm glad you liked the TWIV with Espercalis. It was fun. And I don't know, Amy, if you noticed the thumbnail for that TWIV.
I did.
The name of it was asking Espercalis for some ID, and then I used a black and white photo, kind of like a mugshot thing,
Yeah, I saw.
I don't know. People, maybe they don't appreciate my artistic endeavors. I'm not an artist.
I did. You are an artist. Who tells you that you're not an artist?
Oh, yes, I am an artist of some sort. Yeah. Yeah.
What do you mean of some sort? What is this crapola?
Okay, I'm an artist. I'm also a scientist, right?
Well, what makes you think that they're mutually exclusive? I don't understand.
Anyway, TWIV 1000. Yes, we are going to rapidly.
Going to be in March.
Probably March.
Where are you going to do it? Where are you going to do it?
Well,
You're going to do it from the incubator?
I want everyone to come to the incubator.
Okay.
We could accommodate a few guests who would like to come and sit and watch us.
Yeah, you could have donations for having tickets. Yeah, for sure.
Okay, so that's what the plan is, to have everyone, all the hosts, right? Me, Dixon, Alan, Rich, Kathy, Brianne, and Angela all come to the incubator. You think I can fit them in the room, Amy?
Yeah, but you're going to need David.
Yeah, we need David to come, our video guy, and take care of stuff, because I can't handle it with all those people. Yeah. All right, sure. Reverend Sasquatch. On my first day of symptoms, I did a rapid test. It was positive. Started on Paxlovid, did wonders. Never got more than postnasal. There you go. See? Reverend Sasquatch was not afraid of the COVID rebound. I shouldn't use quotes because that's what it is, COVID rebound. I didn't use the other word. Did you notice, Amy?
I did notice.
What Amy said about rapid antigen tests and treatment, yes, thanks. This has been the experience of others we know, that there could be a delay, right?
Yeah, for sure.
So much misinformation is out there about Paxlovid. You should do a special program on it. By the way, I benefited from Paxlovid after using an antigen test. Thanks for another great year of Twitter. Amy, can I tell them about my planned Paxlovid thingy?
Of course.
All right, so listen up, folks, okay? Stop chatting amongst each other. This is very important.
Okay, well, you don't have a big banner. You didn't have a drum roll. You didn't have anything. Where's the drum roll?
I threw them all out because you said no sound effects.
Well,
let me see
this
here.
One time you can have sound effects. I don't like it. I don't like gratuitous. That's gratuitous use of sound effects.
That's the wrong one. How's that?
No, I want the drum roll for the anticipation, you know?
Oh, I got all these sound effects on my board I never use. Anyway, here we go, folks. You know this
You
company
gotta learn
Pfizer,
where the
they
sound
make
effect
they make
button is.
Paxlovid. So Pfizer would like Daniel and I to do a series of podcasts explaining the science behind it and so forth. And I said I will not take any money for this because I don't
They
want
can
you
make a
guys
big
paying.
donation to Microbe
No,
TV.
I didn't even say that. I think
Well, they should.
anyway. Daniel's not going to take any money either. So is that okay? Or are you going to still? And they will actually say it on each video. They're going to say that we did not provide any reimbursement for this. Do you think that's
That's
cool?
fine. Yeah, I think
Or
it's great.
do you guys think I'll be conflicted? Because I don't want to. I told them I don't want to lose any listener who thinks I'm conflicted. Because if 10
Why
people
are
say
you conflicted? It's your
just
experience. You
if
took
10
the
people
drug.
say I'm being paid to do something, I'm not going to. Even though I'm not, I don't want people. Anyway, let me know in the comments. Okay. But if that didn't happen, yeah, we could do a special program. Yeah. All right. Sandy had a heavy head cold starting Monday, tested Tuesday negative. I have 2 doctor's appointment, an hour and a half away tomorrow. An inspection. I feel good. Would you do a second test? Bless you.
Thank you.
The 2 doctor's appointments are for different things, I assume, right? They're going to want you to test maybe before you go. So you should find out from them. Right?
Yeah.
But it's not a bad idea for your own sanity, too.
Yeah.
You tired, Amy?
Yeah, I get tired easily from being sick, but I feel better. Now I'm sneezing. It's always something.
Lease paid under $2.20 a gallon for gas in Columbus.
Wow, we should
It's
move
cheap.
to Ohio. I don't think I would like Ohio. It's landlocked. I like being close to the water.
5 days after you stop Paxlovid, COVID rebound. Are you infectious? No, you're not infectious. It's an inflammatory phase. You're not infectious. They're wrapped, so he can't drop them and cuss on the air. Yeah, see, they're wrapped. It's true. It's true. I read something last week
Okay, I'm listening.
about a preprint for single-dose PNP hydrogel subunit vaccines for COVID. I think they're supposed to be slow release and long-lasting.
Okay.
Do you know anything about them? PNP hydrogel COVID vaccine. I have not heard this.
It's news to me.
5 days ago, researchers report development of robust candidate single immunization PNP hydrogel COVID vaccines. BioRxiv.
Cool.
So let's see. Broad and durable humoral responses following single hydrogel immunization of SARS-CoV-2 subunit vaccine. So it is a nanoparticle with multiple copies of the RBD and adjuvants. So they have a single dose administration, and they're looking at antibody responses. So, that's okay, but it's not the whole story, right? It's pretty early on here. They would have to show efficacy in people and durability. So I don't know how long it's going to take, right?
Exactly. But the other thing is, it's not all about antibodies.
That's right. They have to do T cells too.
Yeah.
But I'm glad people are trying different things. That's great, right, Amy? We
Yeah,
like
I think it's
people
good.
experimenting.
For sure. It's excellent.
Thanks for bringing that up. Hey, there's spike hoodies too, right? And they're long sleeve spike shirts. A lot of cool stuff.
Cool.
There are even mugs. Hi,
Cool.
I'm usually busy when the live stream is on, but I download them later. I'm homesick. I'm glad to be able to catch you all. Welcome, TS. Excellent.
I'm sorry that they're sick, but I'm glad to have you.
I totally love it when you say, oh, Madonna. I say that to Amy a lot. Madonna.
Yes,
Amy.
you do.
My first student, Nicola LaMonica, used to say a version of Madonna. I can't repeat it here, but I loved hearing him say that. Charlotte loved the Brazil talk. I feel sorry for them. They just got Paxlovid. At least they got it, right? But they only got 60,000 doses of bivalent vaccine, Amy.
That's not so good.
I would just use an ancestral booster if they want to boost.
I would too, but nonetheless.
How wide is the range of coronaviruses that are affected by the protease inhibitor in Paxlovid? No, it's not just SARS-CoV-2. It's actually SARS-1. It's MERS, not as effectively. It's not as potent. And some of the common cold coronas, some bat coronaviruses, it's quite broad. So would you say it's a conserved protease, Amy?
I would think so.
Yeah.
Sounds like it is.
Can you comment on the article about Canadian immunologist who says individuals who are infected with COVID have many fewer T cells? Well, first,
Many fewer T cells? What does that mean?
Let me
Many
just,
fewer T cells. What do they think? This is HIV?
well, yes, exactly. Remember the report which suggested that the virus was destroying T cells?
I don't know. There's so many of these crappy papers. It's too hard to
Well,
keep
in
track.
fact, it can, in culture, destroy some T cells, but there's no evidence in people that this is happening. So, I don't think that this is a thing, Patricia. If you know what I mean.
Not that worried about it.
I thought he said, "Oh, Maronna." Yeah, Maronna. You could say Madonna or Maron. It's two different things. Yeah.
Really? Because it sounds like the same thing to me, where you're just frustrated with me.
Maronna.
Pretty
I
much,
guess you could.
pretty
Yeah.
much just gives the same message.
Maronna, Madonna. I don't actually know. Somebody out there tell me. There's a lacking in my Italian. Is Maronna a dialectical version of Madonna? I don't know.
A dialectical? What is that a word?
Dialectical?
I don't know. That's what you said. I think it's a dialect version.
Yeah, but dialectical behavior therapy is a thing.
Yeah.
Dialectical
Okay.
is a word. And see,
Okay.
oh, Madonna, frustrated Italian's favorite word. It's right. How are we doing on likes here? Let's see.
I don't know. You have the button. You should tell
We
me.
have 366 users and 272 likes.
Listeners, not users. Watchers, viewers.
Viewers. They're called viewers, yeah.
And 200 and what?
273 likes.
Well, where are the other 90 peoples?
Yeah, give us a like. There are not a lot of you here tonight, so
I
give
don't
us
know.
a like.
365 seems like a good number for right before Christmas.
Hopefully the following week
You
it'll
know,
go up again.
lots of people are traveling tonight. You know, it's big travel day.
Not the virus freaks. No, they don't travel.
Well, sometimes they have no choice.
I know. Just kidding.
Apparently, according to After Babel, it is a regional thing.
Okay. Madonna, Maronna. Okay.
Yeah, and according to Patricia, it just means damn.
Okay. Thank you very much. Les says it takes 100 days or less to make a new vaccine variant. It's been done in 66. It does not include clinical trials. Thank you, Les, for the numbers. And Tom likes that you said that may change as my lab expands. Nice science.
Yeah.
Any virus that Amy studies is an important virus by definition.
That's what I think, but I'm self-serving.
Here we go. Raphael says Peru was one of the worst countries with COVID deaths, and there's not much processed food there. So that's the thing about these associations, right, Amy?
I can make up associations. I choose not to. I feel it's not of any benefit.
We need to have a plan in case of COVID. What do you do if you're in a place where they will not give Paxlovid? Well, you
Not
can
give Paxlovid, I move.
No. If you're on vacation, say, what do you do, Amy? Oh, I could if you live in a place. No, it's not realistic, Amy, to tell people to move.
I call my home doctor and have him send me the Paxlovid.
What if you live in Brazil where there's no Paxlovid until last week?
Oh, that's not a good plan.
You could try monoclonals. You could try.
No, monoclonals don't work.
They don't work anymore. That's right.
Exactly.
It
So
is
you
a problem.
don't got nothing.
I have
You
no
got
answer.
nothing.
Artemis. Hey, that's good. No answer for that. It's a real problem. I agree. Inequitable health care, right?
Yeah, didn't you see that? That's why, like, it's really controversial what Gavi and Seppi are doing, where they're not going to, like, foster administration of the vaccine. They're going to put it in the everyday vaccine pile.
Right.
Didn't you see that article? Didn't I send that article to you? I could have sworn
You
I
did.
sent that article
I
to
think
you.
you did.
I'm pretty sure I sent it to you
Are people that eat broccoli
because I
rather
was
than
so appalled. What? People are what?
are people that eat broccoli rather than French fries more health conscious? Could explain better health. Well, I don't think one thing is going to define it. Maybe they eat well otherwise. I just don't think you can
I
base it
don't know.
on
I
that.
eat both. I eat
I don't eat.
both.
I tend to not eat fries because they're oily but I like potatoes. Potatoes are good.
I don't like potatoes unless they're as a French fry, but I don't eat that many.
I like a nice
I very
roasted
rarely.
potato. You know the little potatoes, you can roast them.
Yeah, I know. And then you douse them in butter and rosemary and
Listen to the
salt.
word Amy used. She said douse them.
Yeah.
Do folks know what a douse is?
It's the douse homogenizer.
I'm going to show them a picture of it
Okay.
because here we go. Here's a douse. Where is it? You can buy it. Screen share. Here we go. There it is.
204 24 euros. That's like
Oh, cut it out.
$80.
Look at, they want to give me a coupon for a douse. Anyway, that's a douse homogenizer. You put this glass thing
There's
comes out and you put the thing in it that you want to homogenize, then you put the glass in. It's very tightly fitting and you go up and down.
Yeah.
Amy's got the sound effects there. Can you do that again, the sound effects?
No. No.
Why not? You just
No
did
more
it.
sound.
Okay.
A moment has passed.
Thank you, David, for your contribution. Merry Christmas. Thank you for
Excuse me.
you're helping us spread science. You might want to get away from the mic when you do that, young lady.
I said, excuse me. There's so much I can go with
I
the
should-
earphones in my head.
All right.
I said, excuse me. Look, I said I was feeling better. I didn't say I was perfect. Now I got postnasal drip from
All right. So Festivus. It's Festivus. See, we got a lot of people saying Festivus. And we have Jerry Seinfeld, right?
I told you it was from a Seinfeld.
Festivus for the rest of us. Is that what he said, Seinfeld? Let me look it up. Festivus.
No. You gotta look it up.
Is that what
I didn't
Seinfeld
really want.
said? Amy, do you need a really ugly sweater? Like a Christmas sweater, red and white and green and all that stuff. You don't need that?
No ugly sweater.
Okay. Heather, 6 pediatric deaths from influenza in our area this month. No info if they were vaccinated. Got my kids flu vax. Would love to know the numbers on how protective this year's flu shot is for kids. It'll be a while before we have those numbers because they have to collate the data on who gets flu and which ones are vaccinated, right? But that will come. But there's a lot of influenza, as you know, this season. So it could be that the vaccine is not a great match. We'll have to see. It wasn't a great match in the Southern Hemisphere last season. Thank you, Robert, for your contribution to Microbe TV and Science Communication. Appreciate it. Festivus for the rest of us. We can't leave Vincent out of our nice comments. You could. I don't mind. Give them all to Amy, but she doesn't have blue glasses. You could wear fake blue glasses, Amy. Don't you have some reading glasses at your office? Is that them? It's good. It's a good look. What are they, reading glasses?
Yeah, from when my eyes get tired.
The problem is that they're the blue thingies, and they look blue in the camera, right?
I don't know. I didn't buy them for that.
They want me to live to 120.
That'd be good. Be interesting.
Who was it? Sally says die young. So who was it that said live fast, die young, and leave a good-looking corpse? Was that like Richard Farina?
I have no idea.
Wasn't it Madonna? Die young, stay pretty. She may have said that, but the original was live fast, die young, and leave a good-looking corpse. Yep.
No idea.
If you want to live a long life, the best strategy is to choose your grandparents very carefully. Yes, it's not
Well,
something
your
we can do.
grandparents lived a long life. Grandpa lived to 90, what, 4?
My grandfather lived to 90, 93-ish, I guess. My
And
grandmother,
grandma?
she died in her 70s, I think. Yeah. They both had heart attacks.
Fast and sweet.
So James Dean apparently said that. Live fast, die young, leave
Well,
a good-looking
he
corpse.
did. He lived fast, died in a car crash young, and left a good looking corpse.
I don't want to live fast and die young, no. I would like to live a longer life and get more done. There you go. P.S. The young pediatric deaths in our area were almost all influenza A. Any idea why flu A is worse?
We have very little flu B.
Yeah, the flu B can be bad as well. It depends. It's overall milder, but the reason we don't know, it has something to do with the genetics of the virus. Maybe it's also some host. As I'm going to say that because I know Amy will say, Vincent, the host matters too.
Host always matters. Host
So
matters more
we
than
just
the
don't know.
virus.
I mean, it's a good question, Heather. We don't know the answer to that.
Host matters more than the virus.
Oh, this one I don't know the answer. Is there a presumed evolutionary predecessor to the adaptive immune system? Did T cellular immunity come before B cells and antibodies? So the only thing I know is that, what is it? Lampreys have an ancestor of our immune system. They have this single chain antibodies or something that looks like an antibody. I don't know if they have T cells. This is a really good question that, the evolution of the immune system. And Brienne's got a lecture on it. You should go listen to that. I don't know. And I really should. I'm going to go look it up, Amy. I'm going to go read about the evolution of the immune system. I think it's quite interesting. So it's my weakness that I don't know this. Yep.
I wouldn't call it a weakness, but okay.
Thank you. Thank you, Amy Rosenfeld, for that nice comment. It's just something I never studied, right?
Right. I don't think it's a weakness. It's just something that you can't study everything in 60 years. You need to,
I'm
leave something
70.
for the next.
I'm
Fine.
69.
You can't stuff everything in 70 years. You need something for the next 70 years.
69. I'm not 70 yet.
Ay, ay.
Don't make me
Fine.
older than I am.
Okay, fine. You can't
Okay.
stuff everything in the first 69 years. You need something to do next year.
Amanda is suffering with long COVID, pins and needles all over my body, spent time in the stroke unit, no answer. Any long COVID answers, help, answers, any hope? No, actually, although Amy sent me a paper today which seems to suggest that people who have a long-term loss of smell, they have T cells persisting in their olfactory epithelium, and there's no virus there. So that could be the mechanism.
At least for that part.
So that part of long COVID, the loss, but the other parts, it's going to be a while, Amanda. But a lot of people are working on it. There's quite a bit of money being given out by the U.S. government. Yeah, not anymore.
A lot of COVID funds have dried up.
Oh, so quasi-species continues. That was the question about the immune system. Like mitochondria thought to be ancient captured bacteria that became symbiotic. I wonder if there's something akin with adaptive immunity. No, there's not that, but there's a evolutionary progression, and sharks are like the oldest animals that have a similar immune system as we do. But then as you go back, you don't have T cells and B cells anymore. But I don't know the details. I think it's a really good question. Do you think it's in Immune, Amy?
No.
Do you think it's in this book? Probably not. I bet not. Let's just look very briefly. This is Les's favorite book by Philip Detmer. Catastrophic Damage. Nope. Rebellion, Civil War, Hostile Takeover. I don't like any of these names. Nothing about evolution of the immune system. All right. I'm going to make that my project to learn about that.
You have too many projects.
One can't have too many projects, no?
Well, you don't finish a project, so you have too many projects.
Are there segmented DNA viruses?
No.
How can you be so certain, Dr. Rosenfeld? I'm just curious because you said that very authoritatively. Hmm?
Look it up.
Look it up. That's a good question. I never got that in all the years of teaching. Are there segmented DNA? I found my own blog post. Why are there segmented DNA viruses? Oh, that's a good question. I love when I get great questions like that. Yeah, apparently there are some segmented DNA viruses, Amy.
Oh, what is one?
Now you ask the hard question, right? Let's
Well,
look
if you're
at
going
it.
to tell me I'm wrong, you got to tell me why I'm wrong.
Okay. Let's look it up. Segmented DNA and RNA viruses can be single or double-stranded with a circular, linear, or segmented arrangement. But which is the DNA viruses? Here we go. Yeah, the Gemini viruses of plants. Circular, segmented. There you go. Multiple pieces. Yep, that's a segment. Good. We found the information. John, thank you for your contribution. I use the tomato paste in the tube, but it is a great ingredient. You have tube tomato paste, Amy?
I
You have tube garlic?
No.
Just tomato paste.
Yes.
Be cool and smash that like button. This live stream is the only time I log into YouTube. So I can chat and smash Vinny's like button. So if you're chatting, you're halfway there. Hey, we're not bad. We're at 382 users and 319 likes. Boy, I would love to get over 400, if not just for brief amount of time, you know, Amy. I have a feeling we're going to answer all the questions tonight. What do you think?
Okay.
Questella, this is the only time I use YouTube. Questella, you said years ago that we'd hit 100,000. Thank you so much for your confidence in us. And you're still coming. And the reason I recognize you is because of your glasses, your tongue sticking out, and your name. I think it's all cool. Thank you, SRR, for your contribution. Amy's the best. I'm
not the best.
Yeah, you're the best. Yes, you are. You're here. They come for you, Amy.
They come for both of us.
So apparently we are one of Malcolm Gladwell's favorite podcasts, TWIV. So that's cool. I'll go on.
That's very cool.
But I want him to come on TWIV. So once I'm on, I will ask him. Thank you, Doreen, for your contribution. I really appreciate your consistent support of our work. Dr. Wilson's anti-vax guest proved the need for a very basic virology primer. Questions revealed lack of understanding of high school biology. Virology 101. Worst, maybe not necessary, convinced antibodies exist. Oh, my God. You know, science is not a belief system, Amy. Did you know? They're facts. You can't have your own facts.
Really?
Yeah.
I think we elected a president that has his own facts.
Yeah, it's not right. You can't in science. You can't have your own facts. Maybe in economics, maybe in politics. I don't know.
Really?
Maybe in even
How can
law.
You have your own facts in any field. I don't understand.
Science, no. We have facts, and you can't have your own. You can have your own opinions. You can have your own hypotheses, but you can't have your own facts. Thank you, Doreen. Really appreciate it. Thank you, Ariel, for your contribution as well. Really nice guys helping us out here. Callie says, is there less pre-symptomatic transmission of COVID than before? My mom is the main caretaker for my grandma. She's super burned out from isolating these past few years to avoid getting COVID. I don't think there's any less than before. No, I don't see any evidence for that. However, many people are vaccinated.
Vaccinated.
And so therefore they're going to get less severe disease. And so I think that's what you need to do, isolating and have a plan to take Paxlovid. Get all your vaccines, have a plan. If you want, wear a mask, but for sure, have a plan. I saw a paper today. Actually, you sent it to me. They modeled the use of masks continuously to fight other respiratory infections. Remember you sent that to me? And they
Yes, I
conclude
remember.
that for a year or two, it will prevent infection. But then you have this big population of non-immune people building, and therefore you're more likely to get infected because the masks aren't 100%, right? So having that seronegative population is going to be a killer of using masks. It's an interesting idea, don't you think?
I do.
As great virologists,
I send these papers for reasons,
You just send them to educate me, right?
Yeah, but also just to have a discussion, have people think in different ways. It's not
Yeah, I
just,
agree.
look at the problem in a different fashion. Like we're all, certain of your co-hosts are all about looking at it in one fashion and they don't ever look at it in the other fashion. But we don't like to point fingers.
Do you listen to The Cure?
I did, but I don't have a favorite song.
Can you name one?
No. But
So,
I would know it if I heard it.
So I have to admit, when I do the treadmill, which I do every other night now, I listen to EDM.
Really? Because I thought you weren't listening to anything when I offered.
I started because I think it makes sense for an hour. You should listen to something. And EDM, electronic dance music, is cool. And I heard this song last night.
Which is?
Molly.
Okay.
I'm looking for Molly. I've been searching everywhere, and I seem to find Molly. It's by Wiz Khalifa. Wiz Khalifa. Maybe it's a drug. I don't know. I bet it is.
I would assume it's the drug.
But
I would assume that they're looking for some Molly, meaning they're looking for the drug.
But anyway, the reason it's Wiz Khalifa, who also did Black and Yellow, which is my mantra. If people say, "How do you pronounce your name?" I say it rhymes with black and yellow, and that's a song by
Okay.
Wiz
Well, when
Khalifa.
did you become the rap artist?
I'm not an artist of any sort except a science and podcast artist, but I like my music.
I'm just teasing you. Just get a grip.
Richard is in the kitchen making almond spice for baking.
What is almond spice for baking? I don't know.
Molly apparently is MDMA. You see, we have so many smart people on this live stream. They know all this pop culture stuff. Street cred.
I don't understand where you've been living underneath the brick for all these years.
I've been living in the incubator, okay? I do a lot of podcasts.
No, you haven't been living in the incubator for all these years. I only built it like a year and a half ago. So for the remaining 76 years, you've been living under a brick. I don't know how I associate with somebody who's been living under a brick.
Quasi-Species says, Debunk the Funk is great, but also make sure to check out Back to the Science by Susan Oliver. Yeah, I would like to get Susan on TWIV. That's what people have recommended. Maybe here. Yeah.
Cool.
Yes, Sandy likes Susan Oliver as well. China infections are on the rise, I guess, with a largely naive population, underperforming vaccines. Bound to happen after they ease their restrictions. Yes, absolutely. Yeah. Okay. Is that a Cure song? I'll stop the world and recombine with you? I'll stop the world. What is it? I'll stop the world?
Yeah, I think so. It's either that or Tears for Fears.
I'll stop the world and melt with you. That's not Cure, right? One hit wonder.
But the Cure or Tears for Fears, it sounds like I'll stop the world.
I'll
stop the world is Tears for Fears, The Cure, or Duran Duran. Don't you know anything?
No, I don't know anything, Amy.
I just like to tease you. Don't take it so serious.
I'm not taking it serious.
Oh, it's Ordinary World by Duran Duran that I like.
If there was an Australian journal of virology, Amy would be a perfect editor. Piss poor is a tickable box for reviewers of Australian science journals.
Really?
Is it
That's
really?
hysterical.
Is it really a tickable
That's
box?
hysterical.
Nah, I don't
I
believe it.
like that. I would like that.
Modern English. Somebody wants me to
Modern
put the.
English is an interest. They're not my favorite band, but it's interesting.
You still have no idea about
Love
iodine.
Song. That's the one that The Cure made famous. Is Love Song.
Iodine. What do you want to know about iodine antiviral activity? Do you know anything about
There's no such thing.
Can you tell us about the deployment of the chickenpox vaccine? My son, born in 1998, got vaxxed twice as a kid. Now, just had a blood test, was told he has no immunity to it, so needs to get it again.
Some people don't respond. Like I don't respond to measles vaccines. I don't have any measles. I don't have long-term measles antibodies.
Yeah, so
Happens. It's all due to your genetics. You can't explain it unless you do a lot of genetic analysis. It's true.
Yeah, I believe you. Anyway, it's a relatively recently developed vaccine, but it's not specific to the vaccine. As Amy said, there are people that don't respond to different vaccines. And fortunately, they test it and they see that. But they didn't test T cells, Amy. Maybe T cells are important, right?
Yeah, exactly. Could be.
Wasn't there a flu vaccine in the 70s that may have caused some injury? Yes, the swine flu vaccine deployed in 1976 called Guillain-Barré in about 400 people in the U.S. Yeah. So that was bless you, that was a flu vaccine that was made against the swine flu strain that never actually took off. It was a mistake to make it. But
Ed Kilborn.
Yes, so that is the other vaccine. Yeah, there are multiple vaccines. And it was clear that people were getting Guillain-Barré. They associated it with the vaccine. It was very clear. Thanks, Polio Pete. Merry Christmas, Happy Hanukkah, Pignoli cookies.
Yeah.
So there's evidence that
we're waiting for these cookies.
Boosters have a cumulative risk as opposed to repeated same risk.
I don't know that there's a cumulative risk.
It's a good question, though.
It is, but I'm going to need a tissue soon, so we need to wrap this up.
Why don't you? We only have a half hour. We have a half hour left. Why don't you?
Oh, so you want to have mucus streaming down my face? That's going to look very attractive.
I want to get through some more of these questions.
Okay, fine.
This is a good question.
I'll try to tell my sickness. Come back in 30 minutes.
Well, you could leave if you want. If you don't want to stay, you can leave.
It's inconveniencing, Vincent.
Oh, Madonna.
Madonna. How can I leave? The whole show is about me.
I know. I forgot.
I don't understand. How could your star leave? The whole show is about me.
Okay, stop. I need to say something. Sticky guys, can you put down this question? The boosters, do they have a cumulative risk or the repeated same risk? I want to find that out. That's a good question. Thank you, Sally. Come back next week. We'll have an answer. Building from Polio Pete's questions, if the vaccines are mRNA-based, do the boosters need to be yearly? Well, no, I don't think we know how. Paul Offit would say, no, don't do a yearly booster. Do 3 or 4 doses and have a plan for an antiviral, for example. There is no reason to have a yearly booster, especially if the ancestral is enough. So I don't think we have enough information. Oh, we're at 399 people. One more, folks. One more person come. Please come and push us over 400. I'd be so happy. Otherwise, I won't sleep tonight. Can someone pull someone in the room and have them log into their YouTube account?
It's crazy.
Are you watching, Amy?
I don't know. I'm busy sneezing and trying not to be disgusting.
Anyway, unstable unicorn, I don't think they need to be yearly. But, the CDC is not doing the right studies to look at this. Gabriel, thank you so much for your contribution to science communication. Really appreciate it. Is Twin Daddy says, is it fair to say that vaccinia causes frequent adverse events, though far less serious than smallpox?
No.
What are you talking about? The vaccinia as the vaccinia vaccine that's used for smallpox? It depends on the vaccine, right? The original Dryvax had a very high side effect. And the one of the two vaccines that can be used for monkeypox also had, I think it says Dryvax, right? Has a high side effect. So some of them do have side, but far less serious than smallpox. Yes, absolutely correct. Absolutely. My 96-year-old mom has COVID, asymptomatic. She has had all her boosters. I asked my brother if they gave her Paxlovid. He will go tomorrow. So get her Paxlovid. Absolutely. She is in the risk group that needs Paxlovid. Do you think it's a good idea to go to Paris for a month if we both had 5 shots? We are both old.
Go to Paris.
Go to Paris. Absolutely. Yeah. 5 shots. You're good.
Just go to Paris.
Well, but you're not going to be able to bring Paxlovid with you. But they may have Paxlovid in France, and you could try and figure out where to get it should you need it. That would be a good thing. What do studies currently say about COVID antigen tests and infectivity? I'm still positive on day 11. I don't think you're shedding any more on day 11, are you, Amy? I think day 7 to 8 is about it. Is that correct?
Well, if you're vaxxed, it's much less.
Yeah, if you're vaccinated, it's much less. But the antigen positivity after a certain day is no longer indicating shedding of infectious virus.
Well, after day 5, it's no longer indicating, but you could be positive for a very long period of time. I'm running into that problem, actually.
Well, we have a new listener from India. Welcome, Science Life. We're here every week on Wednesday. My 6th grade kids requested you make a video tour of the incubator. We watch Two of Us as a Family every week. Actually, we did make a video tour, and my
David Renata.
He's editing it. I have to tell him to get it out.
He's been editing it for like months now.
Yeah, David, you
almost
need to
a
get
whole
it out.
year.
He's probably not listening anymore. Let's get it out.
Yeah, you can tell him. Good luck to you.
My doctor did a degree in fine arts before she went to medical school. Yeah, a lot of people have different backgrounds. They don't have to be scientists. They don't have to be science majors. Zan, thank you for your contribution. I liked hearing about the confetti mice they used to study cell proliferation in the recent immune video. It's pretty cool. Gabriel Victoria. Are the stickers able to go on bumpers? In that case, it'll be going on a little Miata, a nice little car. I don't know. Does anyone put the stickers on a bumper?
Well, 1890 can test it out and report back.
Try it, 1890. If you have a problem, I'll send you some more. No worries, okay? I love giving out stickers. Go to Paris.
Of course. Everybody should go to Paris. I should go to Paris.
You can't now. You have to work.
I always have to work.
If you're not accepting money or donations, okay, please answer my questions.
We're accepting both. Money and
donations. We do. You can go to microbe.tv/contribute. Should I put up a thing?
Our Patreon. Sure. Put it up.
Let me put it at microbe.tv contribute. You can go here and multiple ways you can contribute, and it's tax deductible in the U.S. anyway. I answer your questions all the time, Patricia. I do.
Ow.
I loved Kiara's Twiv episode. Maybe you could have another wonderful and funny debate with Slavoj Zizek about viruses. I don't know who that is. Do you know?
No.
You look like you don't feel well, Amy.
I don't.
John, thank you for your contribution. Dr. Often on the channel, AMA discussed how our extra severe respiratory virus season had previous isolation as a factor. Would you have done anything differently before if given the chance?
I don't understand the question.
Would you have not isolated people so as to let them get infected with RSV and influenza virus so you wouldn't have a bad season now?
Then we'd all be dead of COVID, of SARS-CoV-2.
Yeah, I think you'd have to do the isolation the same. Yeah. Okay, now we have a lot of people answering my question about Paxlovid, and most people say it's fine to do the videos, but make sure they state no one receives compensation, right? No consideration. If you are not paid, I think it's all right. If you do it like any other interview, it would be fine. Don't let them do a sales pitch. Okay. All right, so everybody thinks it's a good thing. Yes, okay. Yeah, everyone seems to like it.
Told you.
Because of Wednesday night's TWIV Q&A and Dr. Griffin, I helped 2 people, my son-in-law and a good friend, telling them to get Paxlovid. That's great. I love to hear those stories. Love to hear those stories. I always listen, but never hit like. Tonight I'm joining in.
Great.
Oh, we have 400 viewers, Amy.
Perfect.
And we have 384 likes. I can't ask for more than that. That's really good, right?
Where are the other 16 people?
Amazon Smile is up to $12.60 as a group. We are, I don't know what this means, 760. What is 760.60?
I don't have any idea.
Wondering where Pfizer would air your interviews. I don't know. The usual place like YouTube, etc. I think that's what they would do.
think on TV.
Are there rapid turnaround PCR tests? Yes. Right, Amy? They're really fast PCR tests. They're not the usual amplification, but they are, yeah.
Mm-hmm.
Listen to this. Pfizer spots will only be listened to by the TWIV crowd. Other than them, people will not believe you did not receive compensation, unfortunately. That's interesting. But nobody else will listen, right? Who knows? I mean,
everybody has a problem. You're never going to make everyone happy.
Do you think wearing a mask... Oh, I got an email this week from NY Cohen. Great. I saw that. It was lovely. Wearing a mask for 10 days if you're exposed is still warranted for vaccinated people.
No.
I think only if you're over 75. Wait, if wearing a mask for 10 days, if you're exposed, I see, to protect other people. I don't think 10 days you should need to do that, no. What's the CDC recommendation, Amy? Do you know offhand?
I don't know.
You know, they have so many details. What to do if you're exposed to COVID-19. If you're exposed, you should have your healthcare provider test you and wear a mask. So day 1, continue precautions for 10 full days, they say.
Okay,
that's what the CDC is saying. I'm not going to say no, because then people will say you're contradicting the CDC. Okay? So wear the mask for what the CDC says. Thank you. I'm such a huge fan of yours. I've tested myself 4 times. I'm still negative. It's not COVID then. It's something else. Yeah. 4 times. I was invited to a family dinner Friday, 9 children, 7 adults. I hope I don't get sick. Have a plan. You have a doc that will give you Paxlovid, right? So I think you're okay. Madonna is the Neapolitan equivalent of Madonna. Thank you. That's great. I love it. Oh, sorry. I called you guys the virus freaks. Yeah. Well, we have 408 viewers and 404 likes. Amy, tonight they responded big time for us.
It's great.
Oh, lots of masks from Seattle to JFK. Really, on the plane? How many? Over 50%, would you say? People were watching Zelensky speak to Congress. That's why they were under 400. Now they're coming here. Maybe it's over, right?
No, no, it's still very important.
All right, someone tell Charlotte how to get the free antigen tests.
You can just go to the government website.
What is the government website? Free antigen
I don't know,
tests.
maybe test.gov.
You get 8 free tests. Yeah, just look it up. Oh,
Could
that's
be
not the
just
right place.
test.gov. I don't know.
COVID.gov. There it is. COVID.gov. So, yeah, not everyone can get Paxlovid, right? You have to have certain kidney functions and so forth to do it. Well, if you can't have a plan, then what? You're screwed, eh? Yeah, because the monoclonals are not working. You could do remdesivir or molnupiravir, but other than that, yeah, there's nothing else to do. Ask any nurse or doc in the ED about lifestyle factors leading to what they see every single night. I can imagine, right? I can imagine the things
Hmm.
that they see and people coming in, right?
What is ED? The emergency
Emergency department.
department?
Yeah, emergency department. Possibly remdesivir available in Brazil. Yeah, that's what I'm saying. You can try other things, but I'm afraid that sometimes you're going to be screwed. Yeah. All right. So some people like you to blow your nose live. And you did say, excuse me, so they're yelling at me now, Amy, okay? You want me to highlight one of those things where they yell at me?
No, I don't.
Because you said they don't
Let's
yell at
move
me.
on. Let's move on.
Have you given thought to the historical value of the collection of TWIVs, updates, and Q&As? Are they not an unprecedented chronicling of events, data, etc.? Yeah, I totally agree. And, I mean, they are archived, right? They're there forever, I hope. But I am going to a meeting next year about this very subject, archiving information for historical purposes. And I'm going to point out our collection, which is huge, right? It's huge, unprecedented. There's nothing like it to this depth. Don't you think, Amy?
For sure.
Imagine the archive of all your recordings discussed and referenced. Very cool. I totally like that idea. Look at this. Plaque assay princess sniffles are always allowed.
That is true. I am the plaque assay princess.
Don't forget.
I
Do you have
even
a
have a
T-shirt?
t-shirt. I do. I have a plaque assay princess t-shirt. How could you not have a plaque assay princess t-shirt? Jeez Louise.
Pfizer's testing a new vaccine candidate that has spike and non-spike conserved regions. Okay, non-spike being what, Amy? Like nucleoprotein?
I don't know.
You think we could find out? Pfizer testing COVID vaccine. Let's see. Oh, every time I go to a page that Google asks me to give it permission for my location, every time I go, kind of a pain. All right, I don't see anything coming up. I'll have to look up that. I don't know anything about it. My grandfather lived to 103. He never drove a car.
My grandmother was 101 and she drove a car. So
I'm not
what's
sure
the
the car is a
difference?
risk factor. Yeah.
Exactly. That's my point. And she was the worst driver. The worst driver in the world.
I hope you'll both live to 120. I hope so.
Thank you.
So if I live to 120, Amy would be 100.
100 and a little less than 100.
You have to do this show without me, Amy. I have to show you how to do it, okay?
Okay. I'll worry about it in 20 years.
The intros seem very important to understand. So glad Amy is studying them. She
The
is.
intros are very important to understand. I like the little intros. They're cute.
How long does a clinical trial take, say, a new formulated COVID vaccine based on existing technology? All right, so the original mRNA clinical trials took 1 year. But now a newer one, if based on existing technology, you wouldn't have to do a full-blown clinical trial. You just have to do a small safety efficacy trial. So it would be much less than a year. Helen says, Dr. Morita said it's looking like the vaccine is a good match. Okay. Well, I don't see the data yet, but I'll be looking for it when they release it. Yeah. If T cells are responsible for smell loss in long COVID, is it autoimmune?
No.
It's not autoimmune, but they're doing something. Obviously, they're doing damage, right, Amy? And the idea would be some therapy, absolutely.
Read the science paper.
Good night, Greenfields. It's in Ireland. Good night. Thanks for coming. Appreciate it. Amy is always right.
Not always, but it's good.
Is not the human genome segmented? Yeah, a chromosome could be viewed as a really big segment. How's that, Amy?
Sure. It's not a virus.
Is there any data on long haulers and taking the COVID booster? No. Which booster are you talking about?
I'm sure there is. We just don't know about it.
Not the bivalent booster, right?
I'm sure that there is. We just don't know about it.
No, the bivalent is too new, Amy. This study that I quoted earlier just came out.
I understand, but I'm sure that they are doing the study and we just don't know what the results are. They haven't published it.
Okay.
I'm sure it's being done.
Well, Sam, when we have it, we'll talk about it. Questella is an OG. That's right. She is. Currently have COVID. My teledoc denied me Paxlovid because she said my 2 rapid antigens were due to my recent booster, and she could tell from my dry cough I had a bacterial infection. Was appalled. That's terrible. That's terrible. If you had 2 positive rapid antigens, that's enough. You fulfill all the other criteria. I'm sorry to hear that. Molly is a drug, Amy. MDMA. Okay. I didn't know. The things I learned on this stream. I thought I was the only person who equated Racaniello with black and yellow. No, I do it all the time. And Amy can tell you, right?
Yeah.
I was far more concerned about severe COVID than any sort of quote-unquote COVID rebound. Tested negative, didn't bother testing again. Good.
Good.
Could you put up the link to the Amy papers again? Sure. I have so many links, I don't know. There it is. There's the Amy papers. Mm-hmm. Modern English. Okay. These things come much later, but we have a crowdsourcing crowd here, which is great. Love it. I'm seeing all these band names going by. Oh my gosh. Why did they take the sugar cube polio off the market, Amy?
Causes VAP.
What is VAP?
Causes vaccine-associated paralytic poliomyelitis.
Yeah, it causes polio.
Vaccine-associated paralytic poliomyelitis. That's why it's taken off the market here in the States, but it is given worldwide.
How do you celebrate Festivus? What do you do? What am I supposed to do?
I have no idea. Bow down to a pole. I have no idea.
Thank you, John, for your Merry Christmas, Happy Hanukkah, Kwanzaa. I've now spent over a year with all of you. I just wanted you to do well in 23. Thank you very much. And we are winding ourselves down to the bottom. Have there been documented cases of measles infection impacting SARS-CoV-2 memory B cells? That's a good question. As you know, measles can wipe out immune memory, so you would think it would. But I haven't seen anybody who has looked at that specifically. It's a good question. When we got the pink sugar cube, my parents impressed on us how amazing it was. I was for a vaccine fan since. Isn't that great?
Yeah.
It's a great thing, right, Amy?
Yeah, for sure.
Poor Amy. We'll get through this in a few more minutes, okay? Just
Sure.
looking to see what else we got here. 10 minutes. Amy, we love you. Get some warm drink and rest. I advised my elderly parents to request Paxlovid. The doctor ignored their question and prescribed antibiotics. Should I push for them to get a new doctor?
For sure.
Yeah, for
This
sure.
one's a dud.
I mean, antibiotics, that's
This
ridiculous.
is a dud. Move on.
Amy, stop yelling. Come on. Just because you're frustrated about it, I understand it's a terrible situation, but we have to say, yes, get a new doctor.
Sure. That's why I'm frustrated. Yeah.
Everything I know, you feel badly also. Everything I know about viruses come from Twiv. I don't know much, but more than I did before. Thank you. Here's one for Amy. Do rhinoantros have receptors in kidney cells or have any antigenic similarity with them?
Yes, they have receptors in kidney cells. However, they do not infect kidney cells for the innate immune response. Restricts their tissue tropism.
Are there any scientists that currently refute Dr. Offit's sentiments towards the necessity for the new booster? I'm sure there are. I mean, Tony Fauci said you should get the booster. The problem is there really isn't any evidence that it's better than the original. So that's the issue. Even if you have someone who purports to be in favor of it, it's not really well supported. Question, why do you need to work with Pfizer on the videos? You do great videos on your own. What does Pfizer add to the mix? I think it's a good question.
Greater distribution.
Yeah, I mean, more people will hear it than if we just push it out, right?
Mm-hmm.
Here, if they work with Pfizer, many will believe they are legit, sadly. They already think Twiv is establishment. That's the anti-vax crowd. We are not establishment. That's for sure, right? All right. We've reached the end of the questions. Maybe we've reached the end of the questions, and I will let you go 8 minutes earlier. Happy holidays, everybody. We'll be back next Wednesday. Thank you, Amy, for joining us tonight. What do I mean joining us? For your show. It's your show, Q&A with A&V. Thank you, Amy.
Yes.
Happy Hanukkah.
That's good.
Thank you to our moderators. Actually, to our moderators. We had Les and Steph and Tom and Vanity Nutrition. Oh, at Festivus dinner, families participate in airing of grievances. Oh, gosh, I don't want to do that. Yes, there's a Verb Pack meeting on the 26th to discuss COVID vaccines, including Novavax, if you're wondering about that.
Well,
thanks, everybody, for coming. Have a great Hanukkah. Have a great Christmas for those who celebrated. And we will be back next Wednesday at 8 p.m. Bring your questions. Bring your friends. We did bump 400, so I'm very happy. Thanks, everybody.
cool.
See you next week. Good night.